joining. Let's begin by quickly discussing what is IRIS. IRIS, as you know, is the Initiative for Research and Innovation in STEM. It is a platform which provides you with an opportunity to get recognized on the world stage. It inspires research and innovation in the field of science, and it guides and mentors you to improve your research-based project. So some of you may be new to this platform, some of you are old participants, and all of you know that this is a platform which helps encourage innovation and research and guides you along this path. Request to everybody, please keep your microphone on mute. Thank you. May I have the next slide, please, Shrey? The key objectives of IRIS are to stimulate young minds to take active interest in science and technology, and to encourage students to use research as a tool for creating new knowledge. We also encourage you to use a lot of design thinking in whatever project that you undertake to do. We ask you to be observant, to question, to research, and then ideate, and then go ahead with forming projects which will be beneficial to society as a whole. Next slide, please. The annual IRIS timeline goes like this. And at present, we are at that stage of the year when students are normally working on their projects. This is a time when students can actually sit down, put their projects together, and start writing out the synopsis. The IRIS website will be open to synopsis sometime towards the end of July, and you can then go online and submit your synopsis. The synopsis, of course, we will be discussing that in detail, and we will be guiding you on how the synopsis needs to be written. We hope to have the IRIS National Fair sometime either in December or Jan of next year, as things progress. Next slide, please. So, please pay close attention to this slide, because I know that we have some people who are not from India who have also registered for this webinar. We have to say it is actually open to students of Indian origin who are studying in schools based in India. To participate in IRIS, you need to be of Indian origin, and you need to be a bona fide student of a school based in India. You can participate in IRIS individually or in a team of 2 students. There are 2 categories: the senior category, which is 9 to 12, and winners in the senior category can go on to compete at international platforms such as the International Science and Engineering Fair. And you also have the junior category, which is standards 5 to 8. And students from the junior category qualify for awards such as the Broadcom Masters Award. Now, students who have completed their class 12 board exams at the time of project submission will not be permitted to participate. In the past, we've had a number of students whom we know have finished their class 12 board exams and who have in previous years registered as bona fide class 12 students. These students have suddenly come back to us the next year claiming that they are reappearing for class 12 or they are part of an open school. That will not be permitted, because if you've completed your class 12 board exam in one single year, you cannot come back claiming to be a class 12 board exam student the next year. So if you have completed your class 12 board exams in that particular year, you are not eligible to participate in the IRIS National Fair. I hope this makes it very clear. Again, there are students who say that they are taking a gap year after class 12. Those students, I'm sorry, are not permitted to take part in the IRIS National Fair. You have to be a bona fide student of a school in India at the time of participation in the IRIS National Fair. Can I have the next slide, please? So, Even if you do not participate in the IRIS National Fair to compete, there are ways in which you can be associated with IRIS. You can definitely get in touch with us for volunteering, mentoring, or collaborating with us. If you are a student who is in your undergraduate, doing your undergraduate degree, or somebody who would wish to volunteer, you are more than welcome. Please let us know, send us an email, and we will get back in touch with you. If you are a teacher or somebody who wishes to mentor students, again, please get in touch with us. We will definitely be very happy to connect you with students who would require mentoring. And of course, you can mentor students in your own area, in your own town, and get back in touch with us. To collaborate with IRIS, when I say collaborating, we are addressing people who have institutions wherein you have lab facilities, etc., which could help IRIS students to actually come and continue or come and go ahead with their research in your facilities. If you have a facility like that and you would like to get in touch with us, please feel free to write to us, and we will definitely inform IRIS participants that your lab or your facility is available for them to carry out their research. Next slide, please. And now, without further ado, it is my pleasure to welcome Dr. Kishore Amin, Senior Scientist at ACTREC Mumbai, who is one of our oldest members of the Scientific IRIS Reviews, IRIS Scientific Review Committee. In fact, he has been with the project right from 1999. Dr. Amin is the ex-chairperson of IRIS. He has handled and mentored and judged several IRIS projects for the last 15 years. Dr. Amin has more than 30 years research experience in cancer immunology and is currently working as a senior scientist at Hospital, Department of Atomic Energy, Government of India, Mumbai. He has been a scientific collaborator and a contributor of bringing in flow cytometry technology from Florida, USA. He is a co-investigator in the International Cancer Genome Consortium, a project supported by Department of Biotechnology, Government of India, and he is working in oral cancer with tobacco-related habits. He has shared research findings in USA, China, Italy, and many other forums. We are indeed privileged to have you with us here today, sir. A very warm welcome. And I hand over the proceedings of this webinar to you now. Thank you, sir.
Thank you. Hello.
Good afternoon, sir.
Good afternoon.
Sir, you're audible. Please go ahead.
Yeah, okay,
yes, sir.
Sure.
Yes, sir. You're audible. Please go ahead, sir.
Thank you very much, Sharon, Shri, and Shashank for arranging this IRIS webinar and to take you to the entire subject, that is biomedical and health sciences. Maybe we will find it easy at the end of this my talk, and I'll make it as simple as possible, because health is wealth. And not only that, in case of biomedical, we have got a lot of issues nowadays. You must be knowing about the COVID-19. So that is also an issue by itself. And we know that lots of people are now, in the case of research line, who are senior scientists trying to find out a solution, maybe in case of how to combat this particular dreadful disease. So biomedical comes under different subject category, that is biomedical and health sciences. There are 5 different subcategories, and the first one is cell, organ, and system physiology. The second one is genetics and molecular biology of diseases. The third one is immunology, and the fourth one is nutrition and natural products, and the fifth one is pathophysiology. So you can see these small bit of, I would say, I've given some figures there. Cell, organ, and system physiology, it's a hard science. So that is to deal with all the human beings with, with the different type of, so I'll be explaining to you what is cell, organ, and systems, and how we can try to do research on this. And the second aspect, you can see a ladder-like figure, which is nothing but the DNA structure. So genetics is all about to talk about this particular genetic material. Immunology is another interesting subject, which is considered to be the soldiers of the body, which is under immunology, where you can see the immune cells producing antibody. And the figure which you can see, a Y-shaped figure in the third is an antibody. So we will be discussing about immunology as well. And the fourth aspect is nutrition and natural products. It has nothing to do with the human body, but everything to do with the human body. That is, in case of when you try to eat some good food and then build up. So our body cells take up, and that is how we grow from one cell to all these multicellular organisms. And then in case if the food stuff, or maybe in the case of the environment, is having some issues or problems, then starts the pathophysiology. So all these 5 different subjects, subcategories are interlinked, and I'm going to tell you a story one after another. And in my next slide, please. So I always, when I used to give lectures about iris, I generally feel that some of the students may be bored about science, but some of them may be interested. So I start this particular story saying that once upon a time. So when we talk about once upon a time, it is a story, a grandfather's story. Of course, I being a grandfather, I'm just going to tell you a story about how this particular Earth began. When it said the galaxies, you know that we belong to the Milky Way galaxy, and there are many millions of such galaxies in this particular universe. So we are lucky to have one star, which is called the sun, which is in the solar system. And we belong to this particular family, the solar system. And we are fortunate to have the third planet, which is called the Earth. Of course, everybody knows about it. So I'm talking to you. So whatever you know and I know, so that it becomes easy. So now, in this particular Earth, this is the only planet which is called the blue planet, or the life on this planet originated. So the origination of, the origin of this particular life was from water, that is H2O. And the water started when it said with the different chemical combinations of CHOP. CHOP stands for carbon, hydrogen, oxygen, phosphorus, nitrogen, and sulfur. So this started with the earlier life, which is called the proteins, and these proteins came together to form what is called as a single cell organism. So the single cell organism, where you have an example, of course, you people are all students, maybe in biology, you must have learned what is called as an amoeba, which is a single cell. So there are many such examples. I will not go into the details of it, but at the same time, to make you understand, amoeba is a single cell organism, and it can do all the activities which we people or the multi-cell organism can do: locomotion, eat food, when it's have food, and divide. So this is the genesis of this multi-cell organism. So the single cell organism came together to form what is called as multi-cell organisms. And here we have the invertebrates. So the invertebrates don't have a backbone. And as well as the invertebrates, when you try to talk about, there are 9 different phyla. So I will not discuss about the phyla. The phyla, for example, it is Protozoa, Nematoda, Mollusca, Echinodermata, Arthropoda. These are some of the phyla which one will remember, and it is important that you have to remember. And from the invertebrates, and later we have the vertebrates which have got a backbone. And from these vertebrates, we have 5 different classes of animals, and these 5 classes are I just put an abbreviation, if you can see, PARAM. P stands for class Pisces, A for amphibia, and R for reptilia, A for aves, and M for mammalia. I will not give you any examples, but I will just try to highlight. M is for mammalia, that is in case of the mammalian tissue, meaning say the human being who has evolved from the Neanderthal man to the Homo sapiens. So we human beings, or the human beings, are made up of one, we call the body, and this body is made up of different systems, and the systems are made up of different organs, and organs are made up of tissues, and tissues are made up of cells, and cells are ultimately made up of the nucleus and the cytoplasm. And inside the nucleus is the DNA, which is the genetic material, which is a life-giving material. And this particular DNA is made up of ATCG, that is adenine, thymine, cytosine, and guanine, which is a genetic material. And these are the 4 different nucleotides, ATCG, where the entire life on the surface of this earth, or maybe underneath, are made up of. So ATCG is a life-giving a molecule, which is a nucleotide. And because of this, we are all living. So in case if ATCG, if there is some problem or issues, then we will discuss about that. And that is what is called as mutations. So this mutations can be of 2, one which will be favorable, and this favorable mutations can lead up to forming of new species. And if it is unfavorable, people, they will die. So I have color coded number 1, tissue and organ, which I will be discussing about the cell, organ, and system physiology. And the second one, which is in red, ATCG and DNA molecule, which will be talk about the genetics and molecular biology of disease. And the third thing, which is about the cell, which you can see in the middle there. And that is, we will talk about immunology, how the soldiers of the body tries to protect us during this, and what we call as immunity. How does it help us to maintain our body, against this particular disease, and different types of defense mechanism, which we will be discussing. And in the case of the 4th one, of course, the 4th one is nutrition and natural products. So this is actually the odd man out. If you can see that nutrition doesn't have to do anything inside the body, but it is outside the body, which we have to take it and say we have to do the feeding, as well as these are some of the nutrition habits which we'll be trying to talk about. And this is a very, very important. Without food, we cannot try to grow, or we cannot try to live. So I'll be talking about that also. And the last one is about the pathophysiology. You can see that it is the one which I've colored it as green color, the body. If the body doesn't like this particular system, the food or whatever, so the entire system fails, and then we get disease. So instead of having an easy life, and it will be at disease. So all this corresponds to 1 minute, the earlier slide, please. So all this corresponds to the man who, the brain, which you can see there, the human brain, with the help of a pen as well as a book. Because of this developed brain, we human beings can do a lot of good things when we try to explore. And this is how we try to do research activity in the modern times. So in the next slide, please, I will be trying to talk about how we can try to develop a research-based project. And of course, you must have heard many a times, and some of you must have also dealt with, you must be the finalist somewhere or the other, or in the school, and as well as in your college, you must have done several experiments. But this is entirely to do with biomedical and health sciences. This is something which we have to be very careful while doing experiment. And of course, it's very easy, and many of our students have gone to the national as well as international level. So what are the basic steps which these students have done? So what they have done, the story goes on like this. So in the beginning, what they have done is that they have identified, developed a topic which is needed. Meaning, say there is an hypothesis which is there. So this was a challenging part of research. Now, for the modern times, I mean, say in the recent times, you must be knowing this particular dreadful, the COVID-19. So this was a very new as well as a novel um, uh, virus which has come into existence, and we don't know how the etiology of this, but yet it was a challenging part of the research which the modern times they do. So do a preliminary search for information. Of course, here in this particular, there is no such information about this particular virus, but at the same time, we know that a preliminary search of the other disease which has come earlier by virus, they have some information, so they have collected, and because of that information they have tried to give some medicines which can help, or it is all on an experimental stage. Of course, we have to keep on trying till a particular vaccine is being made or something like that. So a preliminary search for information in the case of biomedical research or health sciences has to be done. And once we have done that, there should be materials and methods. What are the materials which we are trying to collect, and how can this be done, and the different methodologies? When you try to talk about the materials, it can be in the case of, we can try to consider in the case of materials which are related to certain chemicals. It can happen that there may be certain instruments which are needed. Some electron microscope is needed to see a particular cell and how it tries. So a confocal microscopy may be needed, and how we can try to detect. So climbing an Everest is not so easy, but you have to lay a path, you have to see a designated path and then try to do this. So when you climb the Everest, you should know what are the materials which you are trying to carry and how to survive and how to reach the peak. So this is here also in a similar way. We have to have materials and different methods which we can try to solve, or the hypothesis which is there, which we can try to solve. The fourth important step is we have to evaluate our sources and the experiments which can be done and how the results can be interpreted. I mean, say, in case of the sources, we have to, we have to do some experiments, and these experiments cannot be done only once. They have to be repeated at least 3 or 4 times, and then we have to have what is called as a statistical values. And this statistical values should be significant, and they should say that we should try to show it in either in bar diagrams or maybe certain graphs which we can represent. And this is how we can try to represent the results. And once we have done this, all these experiments which we are trying to do should be, should be noted in what is called as a logbook, and everyday logbook has to be maintained. Why? Because we will soon forget what we have done experiments on the during the last week or before that. So that is very, very important to keep a logbook and to write it in your own words and how you try to manage. So this is very important. So making notes, drawing conclusions and references are also equally important, because whenever you take somebody's reference, maybe in the case of certain experiments which has been published, so you have to quote your references, which is important. And the final thing is writing a manuscript. And if you have to appear, or if you have to come for the IRIS fair, then a small synopsis has to be written, about 250 words. And I will explain to you what is the synopsis for those people who are new to this particular word, synopsis. It's also called a synopsis, okay? And then we have the 7th point, which is a full, proofreading, where we have to read the manuscript, and then once it is given, a proper shape, and then a particular hypothesis, it is proved, And this particular proof of the hypothesis could be published in very peer-reviewed journals and things like that, so that this becomes an additional information for the scientific community or the scientific fraternity, so that one more step towards research. In the next one, please. So the question is that students always ask us, when we ask, we have finished this particular experiment, that fellow has done this, and how and where are the questions when it's how to conduct experiments, and what is to be kept in mind while writing good synopsis is very, very important. So people, first of all, let me say that how and where to conduct experiments. These are the questions generally a student will ask, because this is all related to biomedical and health sciences. So something to do with medicine, or something to do with maybe finding a drug, or finding a medicine, or finding a cure, or finding out what can be the outcome, the symptoms, etc. So all these new things can be studied in this particular. The question is how. So experiments to be conducted with supervision of mentors. So a mentor can be your science teacher, or any subject category teacher, or it can be your elder brother, or somebody who can try to help you and who knows much more than you, who can try to hold your hand while doing experiments. And it can be a health personnel, especially in the case of doctors, nurses, as well as healthcare workers, can also try to give, give the necessary supervision which you want. And before doing experiments, what is more important and vital is that a consent form, you should ask the consent of that particular individual or of that particular person when you're trying to work on maybe a survey related science, wherein you have to take the consent of certain people. And then when you're trying to do some experiments with a group of people, there should be sufficient number of subject to be enrolled. So this is in the case of survey-based, where the consent has to be done, where certain experiments can be done in the case of if you are trying to give. But generally, we don't encourage people to do experiments on human beings. It is first what we have to do is that there is a particular phase 1, phase 2, phase 3 trials, and before that animal experiments has to be done, a control and experimental group has to be done. This should be, before starting this, an ethical clearance has to be taken by the institutional ethics committee, which is very, very important. Without that, things can go wrong. And essentially, animal experiments. We had a similar case once upon a time where during maybe about 5 to 6 years back, we had an animal ethics. The pet, these people had raised objection because we had to take some permission, and animal experimentation has to be taken. We should not try to pain the animal. We should not try to kill the animal, and as well as possible, we have to avoid animal experiments, and permissions has to be taken under controlled guidance. And in case of this particular experiment where you have a control group, a placebo can be also used instead of using a drug in the control group. What is a placebo? Placebo is just a small tablet, just like a sugar, which is not the drug, but it is given in place of a drug, and that is used as a control. So many of the students earlier in the last few years, they had made several kits, diagnostic kits, which they won a lot of accolades. And I'm happy that they have been thinking about how good it is to do some research on biomedical and health sciences. So this is one of the vital things. And now the question, where we have asked question how to do the research, we have got the next question is where to conduct this research. So as in the beginning, Sharon pointed out that we can conduct experiments in recognized school labs or research centers where we try to take a tie-up of this particular research labs, as well as certain labs which can help to take students. And of course, the students will not be allowed to touch the instrument, but at the same time, queries, as well as in the case of handling certain things, can be allowed. For example, in the case of an electron microscope, students will not be allowed to operate. There will be special, the technicians, technical hands, so that they will operate these machines. Even in the case of flow cytometry, there are people who will, but we can try to collaborate and get the things done for getting some, getting some results out of it. So now the question is how and where to conduct experiment is over. Now, how do we write a synopsis, and that to an effective synopsis? And I told you, in some of these students who have just joined this webinar, they may be not knowing what is a synopsis. Of course, many of you may be knowing. For those few who don't know, I'm just telling you, the synopsis is nothing but a summary of a planned project which is submitted for approval from competent authorities. So that is just a summary. For example, in case if you go for a movie and your friend is asking you, what is the story? You don't have to tell all the 2.5 hour story. You will just tell him that finally this fellow died. Meaning, say within 2-3 sentences you finish. So that is what is called as a synopsis. Synopsis is just giving a panoramic view of research for quick analysis by reviewers. So research, say whatever you have done. So the synopsis, the body of the synopsis will have a title and the statement of the problem, or what is called as an hypothesis. It will also have aims and objectives, meaning say what you are trying to plan and what will be the objective. And then once you have this, you have a little bit of review of literature, meaning some references, and how you will try to crack this particular research by giving some methodology, by giving a route, by giving a particular solution to this particular problem. And then we have whatever, wherever you have taken the stuff, means that you've taken some of the tips as well as some figures. You have to have references, which is very, very important. So the problem here is that how and where to conduct experiments. I've told you how you have to conduct, and experiments have to be conducted, because this is a biomedical. There should be an ethical things which will be felt, and a health sciences which deals with a lot of other things which are very important. And how to write a synopsis, I've told you, it should be a gist which is not more than 250 words, and this should be sent to IRIS National Fair online, and a group of SRC members will try to check, and they will try to select this particular synopsis. A synopsis will be just like giving a sampling of your entire project. So in the next slide, please. So this is also another important issue, because we are trying to talk about biomedical and health sciences in case of human beings. In case of we humans, we have to see what are the ethics issues while doing this. I belong to the Tata Memorial Hospital, wherein a lot of cancer patients who have got oral cancer by having tobacco-related habits. And there are lots of ethical issues when we are trying to collect their samples, when we are trying to collect their tissues, when we are collecting the, maybe certain cases urine as well as saliva. So there are ethical issues. So in the case of when you try to recruit these patients or recruit the people, we should give them the benefits. What are the benefits which will be there? So in our project, when we try to recruit people, we had given them the benefit of certain money just to come by train and something like that, as well as some medicines were given free, and that is how they could try to have certain benefits. And we should see that that any of this particular medicine should not harm them. It should be non-harming, and they should be having the right to say that to be in this particular system, they have to. So a consent has to be taken, and we should be honest enough to tell them what would be the consequences, or what likely could be the consequences of a particular drug and medicines which are there. And sometimes it happens when a person has got cancer, or he has got certain this one where the prognosis is very, very low. So a confidentiality has to be kept. When you say we have to be we should keep the things secret, not to tell, because there may be some of the patients may be carriers, and in case if a sister is negative, or if the other sister is positive for cancer, then we have to say that these things have to be there. And ultimately, our main aim is to preserve life, but it's a human life, and it's very, very important. And we have to see that the ethics looks after this, and takes care of all the important points which we have to take in. And then proper justice has to be given to the patients, or whoever we have to. So in the next one, please, we have the entire thing can be we have the different subject categories under biomedical and health sciences. The important focus is that we are trying to talk about only human health, and if the health is not there, then what comes is the disease. So the first thing which we have to know is that what type of research you can try to do in case of human health and research. This is very, very important. So the three things which one can do is either make a diagnostic kit, where some of the students have done earlier. And in the second case is in the case of treatment, where you need a big, a lot of people who can try to, it should be a collaborative project. And third is in the case of prevention. So these are the three main aspects of a disease control. If a disease is there, then how to diagnose. Second thing is that if a disease has already come to a person, then how to treat it. And the third thing is prevention. So these are the three things which are now, you can try to see it in COVID-19, where we have to have a diagnosis. In case of diagnosis, we say that if a running nose is there, or a sore throat is there, or maybe a cough and cold is there, so we have to diagnose. And then there's a particular test which can be done by ELISA or PCR, that is a polymerase chain reaction, and then try to say whether the person is positive or negative. And then the treatment, of course, the line of treatment, it has never been, so successful, but at the same time, the young ones who are having a lot of, immunity, and the immune system is working fine. So all that comes under the health sciences, which I'll be trying to talk about. And the third aspect is study of normal functioning and investigate internal as well as external factor, feedback mechanism, stress and environment. Environmental is also important for impact of health. So always we generally say that health is wealth, because why we are trying to tell you that is that we can try to be without food, without water for maybe a few hours or few days, but at the same time, the environment, which is the air and which we breathe, you cannot try to postpone it. Maybe for a minute or two also, it's not possible. So we have to breathe right from the birth to the time when we are not going to live, or we have to breathe. So this is a continuous process. In the next slide, please, we will try to talk about each of these subcategories. So the first one is cell, organ, and system physiology. The second one is this one. So let us try to talk about the first one. In the next slide, please. So in cell, organ, and system physiology, what experiments can you do? What are the interests? So everybody wants to be a doctor, or maybe a scientist, or maybe want to explore something. These are some of the simple subjects categories which has been introduced lately. And I think it is good to learn about this and then try to be involved in trying to find out answers to these questions. So in this particular category, where we have this subcategory, cell, organ, and system physiology. So I told you that a human body is made up of different systems, and the different systems are made up of different organs. Organs are made up of different tissues, and tissues are made up of cell. So cell is considered to be the building block of a human body. So in this particular case, we have to talk about involving in maintaining health or disease. So what we put in through our mouth can lead to a disease if the body doesn't take care of it. Okay, so that is one thing. What are the disease causing? So when when a disease is caused, then we call that as an etiological factor. Etiological means what causes the disease. Etiology factor is what causes the disease. So in case of malaria is caused by a particular mosquito bite, and then there are certain where examples can be given in a number of cases where there is a viral fever, or in the case of a bacterial infection, or whether there is a particular metabolic dysfunction in the case of the human body. So all that could lead to the physiological differences. So here in the second point is that could involve investigating role of cell signaling pathways, intracellular as well as extracellular. So this is also in the case of there is a particular way wherein a cell can be, the nucleus is there, and the nucleus we have the chromosomes, and the chromosomes are the ones where the DNA is there, and the DNA tries to be involved in different pathways, the signaling pathways, etc. So it can be intracellular as well as extracellular, just from the single cell, how we try to give a multicellular, and all these multicellular organisms are controlled by different organs and different tissues in our body. And this is really it is a big network of big network of investigation where we can try to do about the system physiology. In the third one is investigate maintenance of homeostasis. Of course, this is also in the case of hormonal control. So you know that there are hormones which are important. In the case of male, hormones are separate, and the female hormones are separate. So all that is important, and how the hormones during the time or during the age, how it tries to differ, and then it tries to bring in a particular cycle which is regulated. If something happens there, if the protein is not being, if it's not being produced, then the entire balance is lost. So in the last one is that studies in disease which are stress related. Of course, if a person goes on trying to do a lot of work, of course, experiments can be in that also stress related. In case of biochemical, mechanical, physical changes at tissues and organ, which can bring in different types of, stress can bring in disease in case of this. So this is the first part which a cell organ can do, and a number of experiments can be done in this particular category. In the next one, please. We have genetic and molecular biology of disease. Of course, you know that the father of genetics is one who did experiments on the pea plant. And it is very important saying that Gregor Mendel, he was the one who did experiments on a simple, this one without any lab, without any costly instruments, but only on the pea plant. He tries to do experiments and then try to do this. So in the case of genetics and molecular biology, this all deals with the nucleus, and nucleus, its component, which is the DNA, and the DNA and RNA. And in the case of DNA, when you try to talk about DNA, it has got, I told you, the mutations can be of two different. It can be a mutation which can be from the body cells, which is called the somatic mutation, meaning say body cells or the tissues of the body. For example, in the case of breast cancer, the tissues can go, it can, the epithelial tissue lining this particular in the case of breast tissue, it can lead into cancer. and it can be a carcinoma of the breast because the tissue has not divided properly, and then it became carcinogen because of certain components. So the etiological factor can be so many things, the food which we eat and the environment, etc., can lead into this. So the cancer which is formed by the tissues during the course of certain materials which is trying to instigate into the system, into the cell, and then trying to cause. So that is what is called as a somatic mutation, which can bring in, which can bring in cancer. But at the same time, there is another type of cancer, which is the genetic, and this is what is called as a germ line cancer or the germ cell cancer. This can occur in all the cells. All the cells, there will be a defective gene, and this is what is called as a germline. So there are two types, germline cancer and the somatic cancers. So these are the two ways where you can try to study on this case also, trying to take help of cancer specialists as well as scientists who is working in cancer biology. So studies include examining activation and deactivation of genes. So these are also the ways where we can call the epigenetic regulation. Epigenetic means some factors from outside, like in the case of the yellow dye of certain chemicals, which can try to irritate and then bring in. So this particular change or this particular component or this particular chemical is nothing but which brings in epigenetic regulation, or which can try to formulate. So this can happen in the case of skin cancer, where you have this ultraviolet light which comes from the sun, and generally this happens in case of people who are trying to bask in this particular sun or sunbath, which generally happens in the European countries. They get skin cancer or what is called as a melanoma. So studies using this genetic and molecular biology can be of very important. And in case of molecular biology, this PCR, RT-PCR, and so many other molecular technology or techniques are there, so that one can imbibe and then try to study this particular research, which is in category, subcategory 2, which is called the genetics and molecular biology of disease. The next one, please. The third one is immunology. So this is a very hot topic, which is immunology. And you know that immunology is very, very important. This is called the soldiers of the body. The soldiers of the body are very, very important. So we have the external, that is in the case of the border force. So the border is nothing but the skin, the human skin, which will not allow us to I mean, say the invasion of this, I mean, say the bacteria, as well as in the case of cannot be there because the skin takes care of it. And then internally we have the other organs, especially in the case of the spleen and the bone marrow, as well as in the case of the thyroid and certain other organs which are helpful in the immune function. In the case of immune function, you must be knowing that immunity is very, very important. And then if you're trying to know who are the soldiers of our body, we know that it is the white blood corpuscles, which we call the leukocytes. Leuco means white, and cytes means the corpuscles. So RBCs are just used for this particular transfer of hemoglobin, and platelets are used for clotting of blood. But the essential soldiers of the body are white blood corpuscles. So the white blood corpuscles are very, very important. Hello. The white blood corpuscles The white blood corpuscles are hello, hello.
You are audible. Please go ahead.
Yeah. I can't see the screen.
Sir, your video is turned off.
Okay, one sec. No, video is turned off, right? Video, there's no battery.
Sir, please unmute and start. Please unmute, sir.
Hello?
Sir, please go ahead.
Yeah, okay. Yeah, so sorry for the interruption. Of course, I would like to continue with the immune status. I was trying to talk about hello? Yeah. Yes.
Sir, you're audible. Please go ahead.
Yeah, okay, but I can't see the screen. I just wanted the screen. Yeah, yes, please, yes. So in the case of the third part, which is, I said, which is very, very important because of the immunity issues. So our immune system, I was telling you about the skin as well as the different parts of our system, which tries to bring in immunoglobulins to our body. And then these are antibodies which can fight against certain invaders like maybe bacteria or viruses which can try to help in produce an antibody. So immunology is a particular subject wherein people can do research on the aspects like to make monoclonal antibodies. So what is a monoclonal antibody? Monoclonal antibody is a particular antibody which can be made by what is called as a hybridoma technology. So this is also a research-based project where you can try to make liters or maybe a few grams of antibodies using this particular system, and it is all available in different labs which can be done. So these are some of the things. So when I was telling you about all the the immune system, which are the T and the B cells. The T cells generally have lymphokines which are being produced, and the B cells are the ones which tries to produce what is called as antibodies, and all these are important in the case of immune system. Of course, there's a lot many things which one has to know, but at the same time we always say the best cook is that if he knows the ingredients well. So similarly here, if you know the basics of a particular subject well, then you can do good research. For example, I have quoted here, if you want to know something about blood and blood related, you should know everything about what is blood and what are the different types of blood vessels, blood vessels which are there, and what are the different types of components which are there which makes the blood. So in case of blood, RBCs and platelets and the WBCs are there. In the case of WBC, you know that granulocytes which are having granule-like thing, and these cells have our examples are neutrophils, eosinophils, and basophils, and then agranulocytes are lymphocytes, and in the case of lymphocytes as well as monocytes. And you know that all the granulocytes which are neutrophils, they have got this amoeboid type of, they try to engulf the bacteria and any other material, which are coming in the way. But in the case of lymphocytes, they have T and the B lymphocytes. The T lymphocytes produce lymphokine like IL-2, IL-8, and so many other lymphokines. In the case of B cells, these produce what is called as antibodies. So these antibodies could be made by what is called as a hybridoma technology, and we can try to explain this in a better way in the next slide, please. So this is a particular odd man out. It doesn't belong to our system, but it can be attached to a system. And say, in the case of nutrition and natural products, this is the food for thought. Meaning, say the food has to be taken in, and it has to be eaten. It has to be taken in through this system, and then whatever is important nutrients and dietary needs which are important, and this will nourish the system or the body. And we have been growing, the cells will be multiplying, and then from a single cell for this particular human body, and there is a particular limit for the growth, and then this is how we try to get in, and these particular cells from one cell to another cell keeps on dividing and redividing for several hundred times, and then we have what is called as a stem cells, which are also important. And stem cell research also can be done using this particular system. So in case of nutrition, several projects have been have come in, and nutrition in case of people who try to put it in the subject category of botany, as well as in case of the other subject category in case of biochemistry also people put in. So there is a particular close knitting of two subject categories when nutrition and natural products are concerned. In the next one, please. So 1 minute, please. 1 minute. Just the previous slide, please. So here I have just put a small note. Always keep a small notepad with you, always, to write down ideas, work, alerts, and so many things anytime. So this is my habit. I just try to take a small note book or something like that and keep it. Maybe when I'm sleeping also it will be there, instead of taking a telephone and keeping it by the side of the head. A small notebook is important. And then suddenly early in the morning when you try to recollect some ideas or something which you have to do an experiment, and that ideas are bright, but at the same time it should be noted. But so you try to take a paper and a pad and then write down what you have thought about it, so that you can try to experiment. So this is a very important tip I'm giving you. So it is important for you to follow it. And the last, this one is that pathophysiology. So if everything goes on fine, you are hale and hearty. If something somewhere goes off, you have disease. And this is what is called as pathophysiology, because of the physiology fails, and then we have to. So investigations will examine changes in normal physiological balance or homeostasis that causes a reaction within body leading to disease. So this is what we can try to say. It is not the end, but it's the beginning. How pathology, say if a cell is hale and hearty, and then we say that the chromosomes, the telomeres become shorter and shorter as the cell goes on dividing, and then we say the old age strikes in, and this is what the physiology of aging. They say that we bring in dementia and so many other factors which come in, and this is how physiological changes happen in case of from a child to an adult and adult to an old, as you grow on aging. So a lot of experiments or a lot of studies can be done using this particular mechanism of what we can call as a physiology of a human being or human life. Next one, please. So with this 5 points, we have come to this, so when we say something that is always there, that means to say India has won many accolades in different subject category, but I'm trying to highlight that we have one in ISF in the last year and earlier to that also in the same subject category where they have spoken about medical sciences, and we have got, of course, I'm not trying to explain to you about this one. All this is available in this abstract. If you can go to the societyforscience.org, you can get all the subject categories, the award-winning prizes and projects and all that, which Sharon ma'am will tell you or explain to you in a later. In the last slide, which is very, very interesting, is the stepping stone. In the next one, please, the successful steps. So to recapitulate what we have learned and what we have done is that this is a young boy like any one of us. Of course, I'm not young, but you people are young. So first thing is that the first step which you have to take while going home, or now when you are trying to go home, you have to see what an issue and problem can be there. The second aspect is what can be the aim. If there is, of course, people now will say that we are going to do a lot of work on COVID because that is an issue and a problem. Don't venture into that right now, okay? We have got so many other aspects which you have to think about. So aim, object, and the novel ideas. The idea should be novel. Nobody should have done this particular experiment, and it should be important that the particular novelty should be there, and you should know the basics well. The second step, when you climb the second step, you have this research based. Wherever there is, you have got search engines where you have just to try to refer to Google and see if there are anything which has been repeated or they are doing that. But what we can say is that wheel cannot be reinvented, but it can be made to go fast. There can be different shapes of wheels. They can be shapes. So that is what is called as research. So you can try to get something good out of whatever has been done. So this is what we are trying to say that. And you should not drive your car alone in the beginning. There should be a guide for it. So a guide should be there who knows a little, I mean, say much better than you, and then you should try to discuss with the detailing projects, the structure of the projects, and then whatever the records are there has to be mentioned, has to be known by the guide. And the fourth step, which is a very, very interesting step, when you start getting results and then you start smiling with the particular project work, and then there are timelines. You have to maintain a logbook, which is very, very important. And then experiments have to be done 2 or 3 or 4 times. You have to repeat so that you can get a particular statistical value. And then the fifth step is synopsis, a poster, and then you will be a nationalist once this particular synopsis is agreed by the SRC, and there you have the national flag. You become national winner. You can see the flags are flying very high, and then this could lead into Team India, and then you can be one of the ISEF finalists. Okay, with this short talk, I end my session, and I need questions from you, question and answers. Thank you very much.
Thank you, sir. We have a question. A student had raised his hand. Arnav, you may now unmute and ask sir your question.
Hi, sir. My name is Arnav. I would like to ask a question about T cells. So I had done some research earlier about how coronavirus affects our immunity system, right? So I found out that it drastically reduces the number of T cells in our body.
Yeah.
So there's this method. It's called there's this enzyme like immunospot called ELISpot, right? Which helps and detects the number of T cells. And in the previous webinar, I had found out that a fellow student had found a strip, had made a strip of paper containing many chemicals, which could detect the protein deficiency in children, right? So I was thinking, could the similar thing be done to detect the number of T cells? It'll be a easier.
So,
it'll be an easier way to find out if we have corona.
Arnav, you are trying to make a diagnostic kit or something like that, where you can try to detect this coronavirus. And here you have not mentioned T cells. T cells, you are trying to take both the CD4 and CD8 cells, or it is you are trying to have different. Of course, in case of any infection, for example, in the case of AIDS virus also, if it attacks, this particular ratio will be reversed. You must be knowing that. The T cell ratio CD4 to CD8. So this is also a type of infection wherein this is being reversed. So what new thing can happen here? This is my question to you.
Sir, I was thinking because coronavirus, if you see, so many people are asymptomatic, and that's what's the main issue in India, right? So if and T cells, whoever has coronavirus, T cells will be reduced, and that's a fact because that's how the coronavirus will enter the body. So instead of testing, which many people will not be able to do because we have heard so many stories in which they're not accepting asymptomatic people, there could be a kit containing like strip or something, which say you put your saliva or whatever on it, and it could detect if you have T cells, like your number of T cells is normal or like that's an average, which you could refer to and go to the hospital in case.
Okay, so you want to try that, and of course I will not go against you saying that, but if it is feasible, I think you should try. But at the same time, it is unlikely that it can work from saliva because you have to collect a lot of cells, and there should be a particular enriching method where you have the T cells, and then you have to. So a flow cytometer has to be used for trying to see the difference between this and all that. So once the particular T cell is reversed, you want to say that if it is less, then you have got coronavirus infection, right?
No,
so Arnav, I would like to stop you here because this is a public platform and you are giving out a very good idea. So I think it would be prudent if you send in your query directly to Dr. Amin, and Dr. Amin can help you out with that. Okay.
Okay, ma'am.
You can send us. I mean, you can send us an email and on contact@IRIS National Fair, and we will connect you with Dr. Amin. Okay. Thank you, Arnav. Thank you.
Thank you, ma'am. Thank you, sir.
Can we have the other questions, please?
There is a question from there is Poonam Bhatt whose hand is raised. Poonam, I am going to unmute your microphone and you may ask sir your question. Poonam, please ask your question. Your hand was raised.
Yes, can you hear me?
Yes, you're audible, Poonam.
Okay, good evening, sir.
Good evening. Yeah.
All these things. So my question is from nutrition. That we have made a nutrient powder from herbal products. And I want to disclose the name of the item which we have used. And we have given it for testing also to Fair Lab. Then they have given us the nutrient value, which are very good in amount. Now the problem is that whether it is consumable means by the people. So how do we know whether it is consumable, or where should we go for the testing so that we can get the certificate that it is consumable?
See, there are many labs. For example, in case of Mysore, you must be knowing CFTRI. Have you heard about that? Yeah, CFTRI, Central Food and Research Testing Unit. Okay, CFTRI. Okay. You just Google it up, and these people they try to take these types of formulas, and they try to use this, and then they do the testing. So that could be one particular place. CFTRI. Yeah.
They will certify us that it is consumable.
Yes, in case of the consumable. So that can be
thank
there.
You, sir.
Yeah, you're welcome.
Thank
you.
Yeah. There is another participant who has his hand raised. Shashwat, if you could ask sir your question now.
Good evening, ma'am. Good evening, sir. Am I audible to you?
Yeah, very much.
Sir, my name is Ajvika. Sir, I just want to know that, sir, I have read about like in the books that the part of our bodies which we don't use just disappeared after some years, and it has happened many times in the body. So, sir, can I ask you that how does it go and how does it disappear?
What you are trying to say, and I understand, is that a body part which is not being used, you're saying,
Yes, sir.
for many years, it will not be.
Sir, I have studied in a book that for earlier in our ears there is a part which is used to listen the voices which are from very far coming to us, and earlier people used it because there were no phones, etc. things. Now in the book it is written that the part has been disappeared from a body, from the all of the
Okay.
all of
Okay,
body.
okay, okay. So you think it is a boon or a bane? Is it
So
important that you have to listen when you have got lots of noises which is there today, or you want to listen to that minute noise or sound which you feel that it should be there? You're saying that our sensitivity has gone down. You want to say that, right? If you could, if I could understand your question, you are saying that earlier we were very sensitive to that particular sound. Now our ears cannot try to; it's not audible. So
He's trying to say that his question is,
yes,
if
sir.
we are not using one particular part for certain number of years, then will that part actually disappears from the body?
so it is just like saying that use and disuse. For example, if a particular animal stretches up their neck and it can become a giraffe for years, it will take hundreds and thousands of years. It can happen, yes. So
Okay, sir. And sir, but where does it go and how does it disappear? My question is this.
So it is all to do with the genes and genetics. So use and disuse of a particular part or a particular gene, so then it can be there. So that is how the species have evolved, that is Lamarckism. You must be knowing
Yes, sir.
Lamarckism. So these are the things. So
And sir,
it
can
is,
you tell that how can we participate in the webinar competition?
that
So,
Shrey sir will say.
so there is no
Okay,
webinar.
thank you.
So, Shashwat, there is no webinar competition, so to speak. The webinars are happening as of now. These are instructional webinars which are happening according to the subject categories of IRIS. This is not a competition. This is an instructional platform so that you get a chance to listen to experts and you get help in thinking about a project that you can do and research that you could carry out related to problems that you see around you. Thank you.
So there is another person who has raised their hand.
Yeah,
Kalpana,
please.
I will
Please,
now
yeah.
unmute you. Please ask sir your question.
Good evening,
Kalpana?
sir. Am I audible?
Yeah, very much.
Okay, good evening, sir.
Good evening.
Actually, I am a teacher and I am since years working upon such projects and have come and participated into IRIS in the past also.
Very good,
Uh,
wonderful. Very nice to hear that.
Thank you so much, sir.
Yeah.
And we keep on mentoring the students at our school, that's TLF Public School. And what I wanted to ask was that in past years, I have worked upon certain plants, and we worked upon the crude extracts and the decoctions. But we wanted to and have also identified certain components from there which could be useful against uh, maybe corona also. because we have identified those properties, but we are not finding any way to isolate the specific component from there. As we know that we have to go for HPLC or something like that. So right now,
Absolutely.
yeah, so right now in these conditions, we are not able to find out any labs that can be helping us. If you could suggest that. So one part
Yeah.
is this, and the second part is, so right from the extraction to the isolation of the component, and also to test it clinically. Because if I go to a doctor and say that, see, this is the decoction that I have isolated and I want you to use it, is never going to agree to that. So what can be the way, what can be the method to go and test the things clinically?
Okay. Number one, to, yes, you're Kalpana, right? Kalpana.
Yes.
Yeah.
Yes, sir.
Yeah. Kalpana, the thing is good that you have been helping students and then encouraging them for the future of our country. It is wonderful that you have been an instrument in mentoring as well as giving ideas to the students. So your first question, you said that you wanted some lab to work with your component, but what I'm trying to say is that even I, being a scientist, our labs are all closed because of this corona and nobody is doing any work, so you'll have to wait patiently for some time. I don't know which part of India you are in.
I'm in Delhi, sir.
Yeah, so Delhi, I think you have to find out which labs are there, but I don't think you should explore now. Let the things cool down, and then maybe in a month or two, or maybe a little bit more than that, some of the labs can help you. And there are, say, other scientists as well as people who can try to help you out regarding this botany experiments, regarding this plant extract and things like that. I think Shrey or Sharon can help you to find out this. But the second question which you asked is about the extraction, the chemical nature and things like that. That also can be done there simultaneously. It is no issue at all that. And you'll have to wait for some time till you get a green signal. Do you agree with this?
I do agree with this, sir, but the problem is that whatever chemical reagents we are using are very, very costly. And also when we are working with the antigens, or we are working with the animals and trying to create some antibodies and all, because that was a part of my own research. And
Okay.
after my doctorate in microbiology, so that was a part of my research, sir. And I wanted to carry this further, finding out some and like developing some antibodies. But when we talk about these reagents and we look for the labs, actually we do not get an access to there. The reason is that everything is very, very costly. The time of the students over there, scholars, is very, very tough. So earlier I used to go to Lucknow and I used to work with Lucknow University and
Okay.
CMAP. But now it becomes very tough to every time switch on there. If you could suggest me some labs which are actually helping us out in Delhi.
Yeah. In Delhi, I think Dr. Bhatnagar is there. So he is
I have already approached Dr. Bhatnagar, sir, in last few years, and he helped me with certain labs at DU, where I did go and I was wanting to do some extractions and all. But the same problem occurred that professors are busy with meetings and such things, and so I could not get an access there. That was one of
Okay,
the reasons my
I think we'll keep your thing pending, and I think myself and Sharon will look into what solutions can be there, and we'll get back to you as soon as possible. Okay.
Thank you.
Right. Thank you so much, ma'am. Thank you, sir. We now have a video which we would like you to watch. This is regards the participation in the IRIS National Fair. This is from our IRIS and ISF alumni. We will take more questions once the video is done.
Are you ready for the ride of your life? Well, it all begins right here. Your first step is picking a category you can enter by yourself or with a partner. If you are in 5th to 8th standard, you will be in junior category. If you are in 9th to 12th, you are in senior category and have a chance of making it all the way to ISF. Now that you know which category you are in, it is time to find out which subject your project comes under.
At IRIS, you have a variety of 21 subject categories to choose from, ensuring that your project has a category that fits. The categories include animal sciences, behavioral and social sciences, biochemistry, biomedical and health sciences, biomedical engineering, cellular and molecular biology, chemistry, computational biology and bioinformatics, earth and environmental sciences, embedded systems, energy, sustainable materials and design, engineering mechanics, environmental engineering, material sciences, mathematics, microbiology, physics and astronomy, plant sciences, robotics and intelligent machines, system software, and transitional medical sciences.
A display shows the public and the judges what you have done. You're looking to attract and inform visitors about your project. To make it easy for people to access your results by creating a clear, attractive and concise display. The focus should be the work. Take a look at this reference. Your title should be simple and must accurately represent your research. Take photographs or videos of important parts or phases of your experiment to use in your display. Prepare a miniature of your model in case your prototype is too large to be carried along. Make sure your display is logically presented and easy to read. Use neat, colorful headings, charts, and graphs to present your project. Pay special attention to labeling. Be sure to adhere to the size limitations and safety rules when preparing your display.
The IRIS journey begins with registration on the website www.irisnationalfair.org with submission of online synopsis and supporting documents. These entries are prejudged by the IRIS Scientific Review Committee, and shortlisted projects are sent an email inviting them to participate in the IRIS National Science Fair. Winners of the IRIS National Science Fair qualify for an intensive series of mentoring camps and get the opportunity to represent India at platforms such as the Regeneron International Science and Engineering Fair in the United States of America. ICEF is the world's largest pre-college STEM competition. Over 265 Indian students have won over 160 awards at this competition, competing with over 6 million high school students from all over the world each year. Not only are you getting a chance to make your nation proud, but if you're lucky, you could join the 31 Indian students who have had minor planets named after them.
So guys, please don't miss this opportunity.
It's your turn now.
If you're innovative and you really want to bring about a change in the world, I think IRIS is the right place for you.
If you enjoy science and math and computer science and so on, so I mean, it gives you a platform to show what you've done, and you have a chance to go to ICF too.
If science actually kindles your curiosity and you're really passionate about it, IRIS provides you the largest platform in India to show your talent.
Applying to IRIS, not only get to have a great experience, you meet great people and great mentors at that. They're not judges, they're not judging you, they're mentoring you, they're teaching you. They make you learn new things.
In the end, it's not about winning or losing, it's about experience, and IRIS National Fair guarantees great experience. Don't tell people what your dreams are, show what your dreams are through IRIS.
It's once in a lifetime experience.
It's your turn.
It's your turn. It's your turn.
It's your turn.
It's your turn. It's your turn.
It's your turn.
We also have a set of slides which will show you exactly how you can participate in the IRIS National Fair. We'll quickly go through them. And we hope that will answer most of the questions that have come up in the chat. If you have any other questions, then we are happy to take them as well. Shrey, can we have the other slides? Thank you. These are the subject categories which were mentioned even in the AV. There are 21 subject categories. You will see this if you visit the IRIS National Fair website as well. The subject categories which are in gray are the new categories which have been included. We used to have 17 as of last year, and we hope to have 21 as of this year, which is the same number as are offered at the Regeneron Science and Engineering Fair. Next slide, please. Yeah, so the tips on writing a synopsis, I think Dr. Kishore Amin has explained this in so much detail, and we are so thankful to him for that. The synopsis, tips on writing a synopsis, example of an ideal synopsis. We are just putting these slides just for you to browse through them. However, Dr. Amin has taken us through a marvelous presentation today, and he has given you the exact way in which the project needs to be addressed. These are the guidelines for the project display and setup. So if you see, this is what your final project display should look like. Yes, this is very important, and we want to reiterate this again. Students who are allowed to participate in the IRIS National Fair are students of Indian origin who are studying in schools based in India. You can participate individually or in a team of 2 students. You can participate in the junior category or in the senior category. If you're in the junior category, you would be in class 5 to 8. In the senior category, you should be in class 9 to 12. One more very important thing is, students who have completed their class 12 board exams are not permitted to participate in the IRIS National Fair. We have had cases in the past wherein we've had students saying that they've completed their class 12, but they are taking a gap year, or that they are redoing their class 12 exams. We are very sorry, but we will not be able to permit you to participate in the IRIS National Fair if you have completed your class 12 exams once. If you've completed your class 12 exams in the month of March, April, May, June, then you are not permitted to submit your project synopsis in the month of July, August, which is when the project synopsis will begin. Four very simple steps to participate. As you saw that in the AV as well, we'll just quickly go through these slides here. Next slide, please. Once you register, you will find these forms there. Again, the website will open for synopsis submission somewhere in July. We have had a couple of questions asking whether IRIS will happen or will not happen. All that I can say is, with the way things are changing from day to day, we decided to do these webinars because we thought that we should utilize the time as per the IRIS schedule. What happens next is not in any of our control. We hope, definitely hope, that things take a turn for the better and that we are able to follow schedules. If you have any questions, please email us at contact@irisnationalfair.org, and do visit our website, and please follow us on social media.